Okay guys, so we're on lab number 20, which is mechanism of evolution. If you remember when Professor Nelson was doing lab number 19, where we were doing that protein clock theory, and remember she was talking about the different numbers, and she said, okay, it diverged at this time period, and therefore the numbers will be really big if it diverged a lot sooner compared to another couple of animals that diverge way later on, so their number is going to be smaller. Well, let's talk about the diversion part. So what are the mechanisms that cause the evolution, diversion, or for the organism to change over time, which is the definition of evolution, okay? So it's the genetic change in the population over a long period of time. So there are many different hypotheses out there, and what can cause that change or the evolution into two different species. So here we have the different mechanisms. So we have the different mechanisms that can cause that diversion. So we have non-random mating, gene flow, mutation, genetic drift, and natural selection. So we're going to talk about what they are, and then you are going to match it with the correct definition. Okay. So starting with non-random mating. So obviously there's two reproductive partners, okay, and they're mating. So if it's random, that means you mate with whoever. Okay. So let's say I am tall, then generally I would prefer to mate with somebody taller than me. Okay, that would be non-random. I'm choosing my reproductive partner. But if we're doing random, then it's no matter what the height is. It could be somebody who's 5 foot all the way up to 8 foot. Okay, totally random. Alright, so if it's non-random, you are choosing on purpose who you're going to mate with. Alright, so which one of these would go with non-random mating? And hopefully at your end, you pick number 2. Okay. I'm going to make that red and bold so you really see. Okay, did you pick A for number 2? Alright, the next one down, B is gene flow. So gene flow is movement of alleles from one population to another. So in lecture, I talked about 2 different populations of deer. So this population of deer, if you look at the fur coat, would be like a light brown color, tan. But this population of deer has a dark brown color coat, okay, dark brown. So for a long time, they reproduce with each other, and they reproduce with each other. Now, let's say there was a geographical barrier that prevented them from reproducing with each other. Now, that geographical barrier is gone. So remember, from the light brown deer can migrate over and start reproducing with the dark brown deer. So now we're mixing up the allele in that gene pool. So before it was just homozygous dominant. These guys were only homozygous recessive. Now they have migrated over in what we call gene flow. They are now mixing the recessive allele with the dominant allele. So that's what we call gene flow. So which one of these would go with letter B? Okay, so the key word here would be migration. Okay. So number 3 would be B. Thought I made it red and bold. Alright, letter C, mutation. We talked about that lecture. Mutation is a permanent change in the genetic material, and it can be passed on from one offspring to the next. Okay? So the permanent change in the DNA, and can be passed on. That means it's hereditary. So which one would go with mutation? And it's going to be number 1. I want to point out this is the ultimate source of new allele. So a mutation can create, in this particular gene pool, a population of deer, it was light brown, light brown, light brown for years and years and years. And then all of a sudden, in this one generation, we now see different color fur in the deer. And that's what we mean by ultimate source of new allele. There you go. We now have a darker brown color, or maybe an albino color where it's all white. Okay, the next one is genetic drift. A genetic drift is where evolution happened, or a genetic change in a population, totally by chance. It just happened to work out that way. Prior to the genetic drift event, everybody was deer, all the deer was light brown. And then something happened, well no, let me rephrase that. So we had as a population light brown, medium brown, and dark brown. Okay, so we had 3 different shades of fur color in that deer population. And then a genetic drift event happened. Totally by chance, the population's been cut in half. We'll talk about the different things that could have happened to cut the population in half. Okay? So it's just by chance, the half that's gone were all dark brown fur. Just by chance, it just happened that way. So that means what you have left with is going to be the light color and the medium color fur. So that means that every generation after that only have the allele for light brown and medium brown. None of the dark brown, because those are gone. So totally by chance, that's the key word you're looking for. So which one of these will go with letter D? And you should have picked number 5, because it's totally by chance. And last one, E, natural selection. So if you read your lecture outline, where we talk about natural selection. So natural selection is what Charles Darwin came up with to explain how evolution could happen. With natural selection is, in the population of deer, it just so happens where we have an offspring that looks different. And they have some kind of characteristics that makes them a little bit different from the other members of the population. Which just so happened, that characteristic gave the deer an advantage over the others. He had a higher chance of surviving. And because he's able to survive, he or she will be able to reproduce. So if you have a certain adaptive trait, or you have an adaptation that allows that individual to survive, and the members that don't have those adaptations are less likely to survive. Okay? And so because it's able to survive, you will be able to reproduce and pass on the same DNA that has the gene for those same adaptive traits. So the next generation will be able to survive as well. So we're looking for adaptation. So the only one we have left is number 4, is best suited to the specific environment, where they have the adaptation to survive in that population. So number 4 will be letter E. So if you have any questions here, just email your professor. And so now we're moving on to the next part of this lab, where we're going to talk about math. And I know some people may not be math savvy, but it really is simple math. So if you're good at math, no problem. If you're not good at math, pay attention, and I'll do the best I can to teach it. So we have 2 sciences. Hardy and Weinberg, their last names. They got together in 1908 and they came up with what they call Hardy-Weinberg equilibrium. It's two equations that can be used to calculate the gene pool. Okay. So, and remember, in a gene pool, in all of the alleles for that one population. So, like, how many of those members have the dominant alleles? How many of them have the recessive alleles? Okay. They're just trying to figure out a mathematical way of solving for that. Okay, so if there is no evolution going on, that means no changes are going on, then these equations can be used to solve for the frequency of the allele and the frequency of the genotype. So let's look at the two equations up close. So here's the first equation right here is P plus Q add up to 1. Okay. So this is solving for the allele frequency. So in a population of 100 people, okay, in those 100 people you have two alleles. Remember, it could be homozygous dominant, heterozygous, or homozygous recessive. So that means you have 200 alleles among those 100 people. Well, out of those 200 alleles, how many of them are the dominant allele? How many of them are the recessive allele? So when you're looking at the letter P, P will solve for the number of dominant alleles. And then the letter Q will solve for the number of the recessive allele. So if you have 200 alleles, and it's 50-50%, so that means 100 out of those 200 alleles would be the dominant allele. And the other 100 would be the recessive allele, just for an example. Okay. And we'll do some process problem down here, how to use this equation. Okay, so remember, this is for the frequency of the allele, is what you use this equation. Now, we're going to take this equation, we're going to square both sides. And this is how you end up getting that. Okay? So when you square them, you're going to get P squared plus 2PQ plus Q squared. Those three numbers have to add up to 1. So what it's solving for here will be the genotypes. Okay? So up here for P for dominant allele. So if you have P times P, which is P squared, that means you have big A and big A, capital A and capital A. So that means P squared is the homozygous dominant. Okay, big A, big A. So we're solving for, in that population of 100 people, out of those 100 people, how many of them are homozygous dominant? And P squared will solve for that. All right, 2PQ. So remember, P is dominant allele, Q is the recessive allele. So this is going to solve for the heterozygous. So out of that 100 people in your population, how many of them are heterozygous? And then finally, the last one is the Q squared, or Q times Q. Remember, Q is the recessive allele. So Q squared will solve for homozygous recessive. Little a, little a. Okay? So if you understand that, you absolutely have to memorize what each letter stands for. Now this will help you when we do the math down here. Okay? I am by no means a math professor, so if you have any question on how to solve for this, email your professor. Do the best I can here. Okay, so now we're going to use this for our math problems. So procedure 20.1, we have the first question is the frequency of the recessive allele. If it is 0.3, then what would be the frequency of the dominant allele? Okay. So first we got to figure out what the lectures are. What lecture do I use for this equation or to solve for this problem? So the first one that what are we solving for? Is the frequency of the dominant allele. So you go back, remember, dominant allele is going to be the letter P. So in this problem, I am solving for P. Okay. P. Okay. That's what I'm solving for. Everybody understand how I got that? And this is for the dominant allele, and dominant allele is P. Okay. What are we given in the problem? So we're given the recessive allele is 0.3. So what letter is that going to be? And recessive allele is going to be letter Q. The Q is going to be, sorry about that, 0.3. Everybody understand how I got that? But that's given in the problem. The frequency of the recessive allele, which is Q, is 0.3. Okay, so I have this. I want to solve for this. So I have P and I have Q. So remember the equation, P plus Q will add up to 1. Ah. So P plus Q has to add up to 1. Alright, so you know your math, figure this out. Alright, so I'm looking for P, I'm given Q, Q is 0.3, and it has to add up to 1. Okay, so if you're doing a little algebra, rearrange this equation. To solve for P, it's going to be 1 minus 0.3. And you just plug that into your calculator, and you should have for your answer over here, 1 minus 0.3 will be 0.7. Okay, any question on that one? Alright, so that's simple. Now we're going to build up on that. Okay? And the next problem will be a little bit more complicated. Alright, so we're given, if the frequency of the homozygote dominant genotype is 0.49, and we want to know what would be the frequency of the dominant allele. Okay, we already established in this problem up here, frequency of the dominant allele is P, the letter P. So we're trying to solve for P. Okay, you're given in your problem the homozygote dominant genotype. Okay, remember, homozygote dominant genotype is P square. P square. Okay. Okay, I'm gonna get a little creative here. Hold on, give me a minute. Lowercase p. And the key. Alright, so we're solving for P square. And that number is given to you 0.49. 0.49. Ah, so we're going from P square to solve for P. Okay, for the math savvy people, how do we go from P square to just the value, the individual value of P? You will solve for the, hopefully you just that in your head, the square root. Okay? If you find the square root of P square, that will solve for the individual value of P. So basically, you're going to plug in your P square which is 49, sorry, 0.49, then you would do the square root of 0.49. And you plug that into your calculator, the square root of 0.49 would be 0.7. And so I can't do the square root symbol on here, so instead I'm going to do square SQT, square root. And so anytime I do the square root, and this is what I'm talking about, doing the square root. Just button on your calculator, yeah, it says square root on there. Alright, so square root of 0.49 is going to be 0.5. Not a comma, my bad. Alright, everybody understand that? Square root, just like that. Alright, so that's it. My answer over here is 0.7. Highlight that. 0.7. Okay? Now we're going to build up on that. Moving on to the next problem. So, we are given if the frequency of the homozygous recessive genotype is 0.36, and we want to solve for the frequency of the dominant allele. So again, you should already know this by now. Dominant allele is going to be the letter P. It's the letter P. And that's what we're solving for. What we are given in the problem is the homozygous recessive genotype. So remember, the homozygous recessive genotype is Q squared. So we're given Q squared. Square. And that number is 0.36. Okay, so it's going to be a little bit more complicated. Extra step in math here. So we're going from Q squared, and we got to solve for P. Now we already established up here, to be able to solve for P, when you have the letter Q, you use this equation. Okay. So to solve for that, I have P plus Q equal to 1. But my problem here, I want Q, but I'm given Q squared. So I can't just put the number in this equation. That's not the correct answer, or not the correct number. So I have to go from Q squared to just Q. And remember up here, we went from P squared to just P by finding the square root. So we need to find the square root of Q squared to use the Q value in this equation. Okay, so again, I don't have the symbol for square root. I'm going to write out square root of 0.36. Okay. So you plug in your calculator, 0.36, and you hit the square root function, and you should get for your Q 0.6 for your Q. I'm going to go ahead and put that down. Q. Just with your Q, 0.6. I'm going to say that one more time. So we have the square, Q squared, 0.36. I want to go from Q squared to just Q. I need to find the square root of that. So 0.36, and then hit the square root function on your calculator. You will see 0.6 on the screen of the calculator. Okay, you ready to get that? Okay, now ready to use this equation. Okay. So I'm solving for P, and I'm trying to find P plus Q is 0.6. Have to add up to 1. Okay, so you rearrange that equation to solve for P. So P is equal to 1 minus 0.6. Put that in your calculator, and your answer will be 0.4. And that's what you should have as your final answer for P 0.4. Okay? So, if you got this, if you understand what's going on, I want you to take the next minute or two, solve at this when you stop. Okay? Try to solve at this when you stop. Are we ready? When you're not, pause, and then wait till you calculate out your answer. Yeah, your answer is 0.09. Okay, 0.09. Okay, so we're solving for the recessive genotype, so you should have Q squared, okay, and the answer is 0.09. Alright, really quick, how did I get that? So, we are given the P squared. Okay. Remember that is the lecture that we use for the frequency of the homozygous dominant genotype. And that's given to you as 0.49. Then the next step is to find the square root. So the square root of 0.49 is going to be equal to 0.7. Okay, that answer is my p-value. Just my p-value. Alright, once I have that, then I can use the p plus q is equal to 1 to help me solve for my q. So p plus q equal to 1. Okay. So I'm given my value, p is 0.7. 0.7 plus q has to equal to 1. So you type in 1 minus 0.7. So your q value will come out to be 0.3. Okay, you're with me on that one. Okay, to get my q, 0.3 is 1 minus 0.7. Just like what we'd be doing here. Alright, so my final answer is q squared. So that means I have to multiply the q twice. So it's going to be q times q. Right? So it'll be 0.3 times 0.3. Now my very first semester teaching this, I thought my answer was 0.9. And a young lady corrected me and said, no, that's 0.09. I know it's not. I put it in. And she was right. So be careful with that. Double check your math. Okay, so 0.3 times 0.3 is actually going to be 0.09. and that's how we got the final answer for Q squared. Okay? Like I said before, if you have any questions, email your professor. And there's a lot of process. If you go to Google and type in Harding-Weinberg process problems, and you'll get a whole bunch of websites where you can process doing Harding-Weinberg problems. Okay? So we're going to go ahead and move on. Still using Harding-Weinberg, so we'll have plenty of process here. And you should see a table and the data we're going to be collecting. So, trying to get it all lined up. So we have what is called PTC papers. And we would have done those in class, where you would have taken a strip of PC paper. It was there for phenol, thiocarbonate, or something weird word. So it's the flavor that's on this paper. Now some people are born with the allele that allows them to taste this flavor, and some people are not born with the ability to taste the flavor. So I'm going to demonstrate this, and I'm going to take this strip of paper and put it on my tongue. I can't taste it. Okay, I can't taste it. But Professor Nelson's willing to volunteer. So she's coming up here. She said she would taste the paper. So she's going to stand here so you can see her face. And let's see if she is a taster or not.
I am a taster.
So what does it taste like?
It tastes bitter.
Really bitter.
Yeah, a little bit bitter and like chalky at the same time. Some of my students said like soapy, but it goes away. It's not too bad now, like it's fading already. Some people taste right away. Some people kind of like hesitate and then it comes in. So as soon as you taste, you can take it out or whatever. So, hi.
So anyway, so we would go around the room and we would count how many people are tasters and how many people are non-tasters. So here are the numbers that we're going to put down. You still got a lot of water over there. So here are the numbers I'm going to put down. So let's say 21. Yeah, a lot of people are usually tasters.
Mm-hmm.
While the few people, we laugh at y'all, okay. And let's say there were 4. You're going to be that bad. Okay. There were 4 of us could not taste it. Alright, so what we're saying here, if you are a taster, that means you are dominant. So in order for a taster to be able to taste that, you would have to be born either homozygote dominant or heterozygote. You have to be one of those 2 to be a taster. Now these 4 people were blessed not to be a taster. So they could eat bitter food all day long and be like, oh, this is so good. And you'd be like, oh my God, that stuff is so gross. Okay? In order for you not to be able to taste it, you have to be homozygous receptive. That's the only way to be a non-taster. Okay. Now, we're using Harding-Weinberg here. Okay? I'm going to go back, remind you all these lectures. Okay? Those 4 people who were not tasters, they are homozygous receptive. So that means those 4 people would represent Q squared. Okay? The 21 people who were tasters are going to be one of these 2. Remember, some of them could be homozygous dominant, that's P squared. Some will be heterozygote. How many? We don't know at this point. And we don't know. What we do know for sure, those 4 people that are homozygous receptive will equal to Q squared. Okay? But it's not Q squared 4, not 4. And let me show you how you solve for that. To solve for Q squared, put this in red. In this scenario here, to solve for q squared, you would take the number of people that are homozygous recessive, and we have 4, divided by the total, okay? Because you are finding the frequency, okay? What frequency of the population, percentage of a population. So we have to have the total. So it's going to be the number, which is 4, divided by the total, okay? So we put that over here. So it'll be 4, which is the number that are homozygous recessive, divided by the total number of students. So it's 21 plus 4. Do you understand that? Total number of students. So that would be 25. Okay, you understand how I got that? So 4 came from there. The total is these two numbers added together. Alright, so your answer, which is going to be over here. So you type in your calculator, 4 divided by 25, and you will get 0.16. Say that one more time. This answer is 4 divided by 25, and you should get 0.16. Okay, now you're ready to solve for anything that I throw at you on the lab quiz. Okay, once you solve for q squared, then you can solve for any of these letters. Okay, so let's start with q. So remember, q is going to be, q is going to be the square root. Okay? The square root of whatever q squared is. Then you have to calculate them. And so you're going to put in q squared, 0.16, and then hit the square root function, and you should have 0.4. You see that? 0.4. Okay. So how did I get that? One more time. To solve for q is going to be the square root of the q squared value. Q square value solve is 0.16. Put that in your calculator and then hit the square root function. Okay, and you should have 0.4 on your screen. Okay, next one down is P. And remember, P is using that equation, P plus Q has to equal to 1. So to solve for P here will be 1 minus whatever you solve your Q for. Okay? So Q up here is 0.4. So you put in your calculator 1 minus 0.4. You should have on your screen 0.6. And remember, P plus Q has to equal to 1. And you go back up here, and P plus Q has to equal to 1. To solve for P, it would be 1 minus whatever your Q value is, okay? So down here, our Q value is 0.4. And so 1 minus 0.4, and you get 0.6. Okay, to solve for your P. To get this one, all it is P times P. P times P. So we go back up here for your P, we solve it 0.6. Then you put in your calculator 0.6 times 0.6. 0.6 times 0.6. And you should have on your monitor or your screen 0.36. Okay, now this one we have not done. And this is the heterozygous genotype. To be able to solve for your heterozygous genotype, all it is 2 times whatever your p-value is times whatever your q-value is. Okay? So it's going to be 2 times, my p is 0.6. Times q. And q is up here is 0.4. So that's all you have to put into your calculator. Okay? And you should have 0.48. Okay? Alright, so we're going to do this one more time. And I want you to do this yourself. And let me give you the numbers first. So if we were in class, we would go around the room and we would count the number of students that have unattached earlobe. The only way to have unattached earlobe is you have to be dominant. Okay? You have to be born either homozygous dominant or heterozygous. The ones that have attached earlobe, and remember when we talked about in genetics, your whole earlobe is attached to the side of your head. And the only way to have that is you have to be homozygous recessive. So we'll go around the room and we would count, and we counted, let's say there were 15 of y'all that had the unattached earlobes, and 5 that have the attached earlobes. Okay, what happened? Now, so I want you to take, 3 or 4 minutes, however long it takes you, you go ahead, pause the video, and take the time to solve for all of these using the numbers that's given to you. Okay, so if you solve for all your numbers, now let's see, this is what you got. Okay, remember, for your q square, you're going to take the number 5 divided by 20. Remember, it's the number of the homozygous recessive divided by the total. So 15 plus 5 is 20. All right, so it'll be 5 divided by 20, and you should have 0.25. So pat yourself on the back if you got that. So we're off to a great start. This is important. You have to be able to solve for this. Once you have the number, then you can solve for anything that I ask of you on the lab quiz. Okay? Alright, so once you have your q square, and then you can just fill in the rest of the table. So q is going to be the square root of 0.25. Square root of 0.25. And you should have 0.5. For your p, and remember p plus q has to equal to 1. So if q is 0.5, then that means p has to be 0.5 as well. Alright, so p squared is p times p. So if p is 0.5, then to solve for p squared, it's going to be 0.5 times 0.5. And if that's what you did, then your answer should be 0.25 for that one. And then finally, last one, and then we're done doing the math, okay, is to solve for the heterozygous genotype. And it's 2 times p times q. So it's going to be 2 times 0.5 times 0.5. And then you should have, for your final answer, 0.5. Okay, if you got that, pat yourself on the back. If you're still confused, email your professor or go to Google and type in process Harding-Weinberg problems. And the more you do, the better you will get. Okay. Okay, so what does it mean? Okay. What do I learn from solving the numbers? Okay. So in the real world, they would go out and research various populations, and they want to know, is this population of frogs, or this population of deer, or this population of polar bears, okay? Are they evolving over time? Are they changing over time? Okay? So that's where we would use the Harding-Weinberg. The purpose of the Harding-Weinberg is to get a baseline number. So you would go in and you would do your collection of the gene pool, and you do your Harding-Weinberg, and you would have your number. Okay? Let's give an example here. So we are using the mechanism of natural selection that can cause a population to change over time. Remember, natural selection is all about adaptation. Do you have what it takes to survive where you're living? Now, we are looking at a particular population of crickets. They live in Hawaii. So how horrible for the people that have to move to Hawaii to do this research. Okay? So they're in Hawaii and they get the baseline. What they do is they go out and count the number of crickets that are vocal, are able to chirp. Okay? They chirp, 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 chirp. In order for those crickets to chirp, they have to be dominant. Well, there's also a percentage of that population of crickets that do not chirp. They are silent. And so people are like, well, that's not good, because when it comes time to reproduce, they chirp to each other. Okay, and that chirping is what helps attract the female to the male, or help them communicate and find each other in a field. Okay, so they have to have that chirping. So why would a percentage of crickets be born not able to chirp? And how would that be an adaptation? And here's the problem. There is a fly. That's a parasite. Okay? These flies will find those crickets. And when they find a cricket, by listening to their chirping, like, oh, I hear it, and they go find it. And they will land on the back of the cricket and lay their egg under the exoskeleton of that cricket. So, needless to say, when the eggs hatch, the cricket is going to end up dying. So it's not a good thing to be a loud, vocal cricket, because those flies will find them right away. Well, what about the crickets that are silent? Flies can't find them. So they get to survive because they don't get any eggs laid under their exoskeleton. So they get to survive and reproduce. And because they're able to reproduce, they're going to pass on the same allele to the offspring that will make them silent as well. Whereas on the other side, the ones that are chirping, if they got found by the fly and the eggs were laid and they're under the exoskeleton, they end up dying. If they're dead, guess what they don't get to do? They don't get to reproduce. If they're not reproducing, they're not passing on those alleles that allows them to chirp. So what do you think is going to happen over time? Okay, remember, evolution is a process that takes a long period of time. So, here's the baseline. They found in this field of crickets, 500 field crickets. And they used the Harding-Weinberg. They found 25%, or 0.25, or silent crickets. Okay? And then, just like what we did up here, we would solve for all the numbers. So here's their baseline. So when they first started doing the research, they found 25% were homozygous dominant, 50% were heterozygous, and then 25% were recessive. Now they hung around for several generations. Now remember, it is an adaptation to be silent. They're more likely to survive than the ones that are chirping. So if we go back, 50 years later, 100 years later, we'll see what happened to the numbers. So here 25 now is 0. Okay? 50 dropped down to 20, and here's the biggest change right here, 25% being recessive or silent went up to 80 because it's an adaptation. It allows them to be able to survive. So, did evolution happen? And your answer would be yes. Okay, how do I know that? The numbers have changed. Okay? Anytime the numbers have changed compared to the baseline. Okay? What I mean by baseline here is what we first got. And then we went back 50 years later, and here are our new numbers. So these numbers have changed compared to the baseline. So anytime they have changed, the alleles are not the same as they were. So yeah, evolution didn't happen. Okay? There was a genetic change in their population. Okay, that's natural selection. Now we're moving on to the next exercise, which is we were talking about genetic drift. You remember in the beginning of the lab, this will cause evolution totally by chance. Totally by chance. Now, genetic drift can happen one of two ways. It is bottleneck. Okay? It is bottleneck. The key word that you're looking here is death, unfortunately. It's a percentage of the population died out. Okay? They're dead. What caused that? It can be natural. Like when we had Harvey, we had all that massive flooding. So how many deers died because of that? Or squirrels, or birds, the percentage of plants or animals that died from that flooding. Or it can be a tornado, or it could be a forest fire, something that was natural. Or it could be human related, habitat loss, or hunting, overhunting. Okay? So we killed off a certain percentage of the population. Now, what do you have left? The ones that survived. Whatever allele that's there, that's all they have to pass on to the future generation. Okay? So the population will change. It is totally by chance that during Harvey, all of the green frogs died. So all we have left will be brown and gray frogs. That's it. So that means they only have the allele for brown colored skin or the gray colored skin. And when they reproduce to create the next generation, you will only see brown and gray. You won't see any green because the allele that gives the green color, they're gone, they're dead. That makes sense? Okay. The next one is founder. Now this is good. Hey. Nobody dies in this scenario. Okay. Well, I just did this. So in founder is where you had a population, okay, this big, huge population of frogs. And just so happened that a percentage left. It could be voluntarily, they're like, I don't like y'all, peace out, they left. Or they got separated, like the flash flood. And there was a flash flood and it carried away a percentage of frogs down the river. They got separated, okay? So it could be voluntarily or involuntary. So a really good example in humans back in the colonial American time, and when all the people came over from Europe and they set up towns along the east coast of America. And so they would, for a number of years, have this town going. Well, then a group of people say, you know what, I don't like the mayor, or I don't like the church, or I don't like something. I'm going to create my own town. And they would recruit all these people, and they would leave and set up another town elsewhere. Now remember, totally by chance, all the people that left have blonde hair, only blondes. So that means when they go set up and founded another town, and they start reproducing, that means every generation are gonna have what color hair? Blonde. Okay? And remember, totally by chance it just happens that way. Okay? So those are the two different ways when you have genetic drift. So here we have an example. So be sure you understand what they mean. So I give you a little short paragraph of a scenario, and you would have to tell me, is this bottleneck or is it founder? Okay? All right, so let's take a look at the problem. Let's say a group of frogs took a ride down on a log down the river, and they settled in another pond 12 miles away from the main population. Okay, so this would be bottleneck or founder. Remember, the key word to look for is death. If members died, that means bottleneck. If nobody died, it was just separated, that's founder. So the answer to this would be founder effect, okay? All right, so let's go back to the Harding-Weinberg. So we were looking at the frogs, and of course you have to have your baseline, and we got 25%, 50, and 25% for the three different genotypes. And frogs got separated because of founder, and let's see what happened 50 years later. And we can see the numbers are the same. So if the numbers are exactly the same as the baseline, did evolution happen? Hopefully you wrote down on your paper, no, okay? If the numbers are the same as the baseline, that means the gene pool, or the allele frequency, or the genotype frequency, did not change. Did not change. Okay. In other words, evolution did not happen. Okay. All right, did you understand that? And that's all I have for you folks, and that's all I have for you, period. We are done with labs, so yay. So if you have any questions, email your professor. Bye.